So happy Monday. I hope that your new week has gotten to a good start. I can't believe we're already approaching the end of the September. So 27th is the date that we're looking at today. So definitely time has gone fast. What I'm hoping to do today with you guys, first of all, I'm coming down with a cold, so I'm probably gonna take it a little slower and we'll do what we can. But what I'm hoping to do with you guys today is to go through the essential components of the chapter 5, which is all about plasma membranes. So last time that we met, we discussed just a general survey of the most important parts of a cell. And we'll notice that we started with the membrane, cell membrane, just in terms of defining the boundaries of the cell. But after that, we found that many membranes were found within the cell. And it is thanks to these membranes within the cell that allow us to have these specific compartments in a cell and for very specified processes to take place within the cell as well. Since we are going to be moving through material today, probably a little bit in a calmer matter, if I use that word, since I'm not quite 100%. What I would ask you to do is if you could post your name at this point so I can get the attendance. I will ask for that later on if you didn't get a chance to post, but if you can already at this point post your name to the chat, that would be a huge help. And before I jump into the chapter material, I also wanted to acknowledge that I have few messages that I haven't had a chance to answer yet. And again, I apologize. I'm kind of not 100%. I'm a little bit unwell. So if there's anything that's urgent, I will find a way that your case will not be harmed because of the urgency factor and because of me not having had a chance to answer to you immediately. So we'll figure it all out. There's definitely no downside or no harm to your case about the fact that it's taking me a little longer, a couple of days longer than normally to get through materials. But other than those things, does anyone have any questions that are really burning that you want to get addressed so you can focus on the current chapter? If that's the case, just unmute yourself, give me a shout out or wave your hand or write it to the chat, whatever works for you. Otherwise, I'll just move on to the chapter material and try to go through it. I also wanted to thank last time, I think one of you showed me, because the Blackboard has been acting up a little bit, showed me that the screen share that I was doing was hiding a little piece of the information. So I'll try to be very mindful of that doesn't happen this time. But we are back to the same place that where we were before, that once I open up the presentation, unfortunately, I won't be able to see our chat. So if at any point you have questions, just unmute yourself and speak up. It's not that I'm ignoring you if you're typing to the chat. I just can't pull it on the screen at the same time in a way that this Blackboard course is set up. Well, having said all that, let me just jump and share the screen. And really, probably the first thing that I wanted to discuss with you was that should be better, was that when we talk about cell membranes, and we're going to focus on the membrane that separates the inside and outside of the cell. But as you remembered, we saw these membranes also within the cells. But when we talk about these membranes, we end up noticing that there's actually quite a few different terms that are being used. And sometimes it is a little easy to get confused because of that, about what are we really talking about. But all of these terms really would be referring to the same thing. We're just emphasizing different characteristics of the membrane. So that's the only thing that might be a little bit of a throwing off. Why do we have so many different names for the same things? But I do notice, and I acknowledge that sometimes when I'm talking, I'm kind of changing around what term I use. And it is not to confuse you. It's simply to emphasize a specific aspect of that structure. So really, that's all I'm wanting to say. Is a slide. The sentence at the top, you'll see a Finnish sentence and Swedish sentence. It's a saying that's from my native country, and it really refers to the fact that when we have something that we really enjoy, like, we tend to give nicknames for it, whether it's a human being or things. And same thing applies when we're talking about structures when it comes to the membranes in the cell. But the two ones that you probably see me using most are going to be the plasma membrane and cell membrane. So those are the ones that are definitely worth picking up. But you might see me, and I think that during this session that we have today, I'll actually refer to the same thing with all of the names that you see on this particular slide. Your textbook starts this chapter by making a comparison of a cell to the central station's wall, central station in New York City. And it says that the cell membrane could be viewed as the walls of this central station. So if you've ever been to a central station, or if you have seen pictures, videos, movies of there, you'll know that it's a very, very busy site. There's a lot of hustle and bustle going on. And really, the biggest thing is that there's people coming in, people going out, people being directed in a controlled manner from one place to another. And that's really what the analogue with the textbook that you'll be using is trying to make. It's trying to tell you that similar to the walls of the plasma membrane, the walls of the central station don't just separate things. They also help to direct things to the right places. They help us to choose when something should pass and something shouldn't pass. They can encourage us to move to a direction that would be a natural movement, but they can also help us to move to a direction that would not be where we would spontaneously be heading. So really, I think that it is not a bad analogue, but don't let that throw you off. It's really nothing deeper. It's not something groundbreaking that you should get away with that analog. I guess that the biggest message, if I were to highlight anything of it, is that consider the very active and very dynamic nature of a cell. The fact that cells are constantly changing, there's constantly materials moving, similar to this major transportation hub. So I guess that's the biggest take-home message from that. So I've already shared some of these key points when we discuss about the plasma membrane. Remember that the diagrams that you see that can look very much like what you're seeing here, they're not fair to what the plasma membrane and cell membranes really are. Plasma membranes, cell membranes, they're much more active. There's material moving throughout the cell, but also, or throughout the cell membrane, but also within the cell membrane. So none of these structures that you see are stationary and just locked in, as you would see in these pictures. There's much more going on. It's much more dynamic, much more active process. So remember, when you see these diagrams, they're only snapshots in time. They're not really telling you about the great amount of diversity, great amount of movement that we really end up seeing on these cell membranes. And I've listed some of those things here that are essential to note. Do notice that plasma membranes, they are the ones that play key role in choosing what gets to enter to the cell and what gets to leave the cell, and also blocking some of the things from being able to do that. So they are selective. They are making choices all the time, what gets to go through and what does not. So that's the first important key characteristic of a plasma membrane. Another thing that we end up seeing with the plasma membranes is that plasma membranes are constantly changing, and they're also changing in terms of their shape. And the example that I've chosen here is that you've got a picture at the left bottom corner of the screen of a blood vessel that's a tiny, tiny capillary vessel and a red blood cell trying to squeeze through it here. So similarly to the walls of a cell, in this case, the cell is just a red blood cell. It is able to squeeze. It's able to adjust to the changes of the demands that the cell has been exposed to. So cells are rarely as rigid when we talk especially about animal cells as the plant cells are. You remember plant cells had a high amount of materials on their walls that made them very structured and very stationary, whereas animal cells are much more capable of changing their shape and adjusting to the needs based on that. The other thing that we notice is that the plasma membranes play an important role by playing a role in choosing and detecting things around us. So what we're constantly doing in our body, we're screening every cell that's in our body and every other thing that's in our body. That is this part of us, or is this an invader in us? And the reason why we would do that is that whenever we have an invader, typically a pathogen of some kind, it is a risk for our well-being. Many viruses, for example, try to sneak in to the body without being detected by body's protective systems and then cause trouble. Well, the way we deal with viruses is that we try to detect them as soon as they get in. And to do that, we have these little antenna-like structures coming out from the cell membranes and especially from particular proteins there. So they're really doing the screening that are the things around us part of us or not. This also leads, if you think about the organ transplant rejection, if something's not part of us and we've implanted that into the body, your body will now consider it as an invader, as something that shouldn't be in you. So in that case, we need to now manipulate how your body would normally work and how your body would normally reject things that shouldn't be there. So again, it all comes down, even on these very specific examples, to the very basics that we're learning on this course. And final thing that I wanted to discuss about is that cell membranes play a key role in controlling also how messages are transmitted from one cell to another. And here we've seen this picture before. What we're seeing is a plasmaplasmata from one blood cell to another, and there is actually a physical gap for the materials to pass through. And often these would be hormones, for example, that we're passing to pass messages from one part of the body to another part of the body. Well, so far, I think that this all is kind of an introduction to why does it matter and why it is important that we appreciate plasma membranes. I also wanted to take a little bit of time just to kind of discuss how we ended up with our current knowledge about the plasma membranes. But what's important here is that you know that I'm not saying that everything we've done was correct, but actually we were remarkably early on in the history and early on in the research of this topic on the right track of figuring out what's going on and why membranes behave the way they do. So I always think it's worthwhile to look into the history. So plasma membranes, cell membranes, they were first identified sometime in 1890s. So that's during the time when the Robert Hooke talked about those minute pockets, cells, minute particles, minute pocket cells, and how they were separated by plasma membranes from one to another. And we talked about the Robert Hooke, or you looked into the Robert Hooke when you completed the lab on the microscopy last week. Well, what the plasma membrane is made of, that was identified in 1950. And we identified that the main components of plasma membranes are lipids and proteins. We didn't yet quite know how these lipids and proteins are arranged, but we knew that there's a high amount of lipids and proteins in plasma membranes. Well, in 1935, Davidson and Danieli proposed that you could describe a cell membrane a little bit like it would be a railroad track in terms of its appearance. So they told us, and they were able to, based on their research, conclude that the plasma membranes are made of 2 layers, just like sandwich, and then there are things in between. That was not too far from the truth, although they got it mixed up that they thought that lipids were where the proteins are and proteins are where the lipids are. So they were a little confused, but they were definitely on the right track. And as the microscopes developed further, we were able to actually see better of what's going on. And with this development, during the time from 1950s to 1975, we were able to develop much better model of the plasma membrane. And a lot of that work needs to be credited to Singer and Nicholson, and they discussed about this double layer and introduced a term called fluid mosaic. And we'll discuss what I mean when I say that plasma membrane looks a little bit like a fluid mosaic. But what they noticed is that we have these proteins, and they are embedded within 2 layers of phospholipids. So they are essential part of these cell walls, and they are within this kind of a sandwich-like structure that we heard before. This explained both the structure and the function of the plasma membranes incredibly well. So by the 1975, we had a really good understanding of not only what cell membrane was made of, but how it works as well. Well, what I want to do, I want to expand a little bit on this fluid mosaic model that I just mentioned. So anyone who does arts or is interested in things like that will have probably heard about mosaic pieces. Mosaic pieces are these usually made of either ceramic or rock, small pieces that are laid next to each other, and by all these little pieces next to each other, they form a picture. So all of these little pieces become part of something greater. They contribute to the whole that we're going to be able to see. So the term mosaic in this sense refers to the fact that plasma membranes are made of many different things. So we end up finding the phospholipids, which is something that we're going to be focusing on quite a bit, but we also have many different kinds of proteins. We can have cholesterol, carbohydrates, which were those antennas that we talked a little bit about. So many things make up this mosaic of plasma membrane. The other term that we keep hearing used when we discuss about the plasma membranes is the fact that they are very fluid. And by that, I mean that they are constantly changing. So components are moving through the membrane, but also within the membrane. So there's really a lot of fluidity going on. It's far from being stationary and still structure. So that's where this kind of a term fluid mosaic model of plasma membrane comes from. And what I'd like to do now, if you don't have any questions, I'd like us to up the game a little bit, if you wish. And we've discussed the general, what cell membranes are made of. We've discussed a little bit of the history, a little bit how they are much more involved than we might initially think. Now I want to get to the nitty-gritty and look through what is the membrane really made of. But before I get too carried away with that, I just want to make sure, does anyone have any questions? So just unmute yourself and give me a shout out. I'm not hearing anything, so I'm just going to carry on, just because I want to be respectful of your time and not keep you overtime either. So we're going to look at an individual phospholipid next. So I've mentioned a few times now that plasma membranes are made of many phospholipids. But to understand how they end up being packed next to each other, the way they are packed next to each other, we need to first study a little bit of one of those individual ones. And what I've done here, I've drawn a diagram that's really quite a simple diagram showing the two main parts that these phospholipids are made of. So you will see that it has a head part and it has also two tails. And we'll talk about the heads and tail parts of the phospholipid in a greater detail in just a second. But at this point, if nothing else, just notice that it is made of one part that we are going to be calling head, and then two tails that are going to be important. And if we do end up writing down the chemical structure of a phospholipid, we do end up noticing that these parts are even chemically completely separate parts. So we do have the phosphate group, and phosphate group is what gives rise to the head part. Then we have two fatty acid chains, and these are the tail part, or two tails of a phospholipid. And these are connected by a glycerol molecule. The other thing that I should mention when we talk about these fatty acids is that one of these is saturated. So you remember this one had just single bonds. So it's more of a straight in structure when we draw it out. Whereas the other one is going to be unsaturated. And because of just one single double bond, there is going to be this kink in its structure of this phospholipid, this lipid tail that we see on this phospholipid molecule. So really, why I want to highlight that is that all of the concepts that you learned in the basic chemistry come in handy now. We start to move to bigger and bigger scale in the study of life. So I mentioned earlier on about the head and the tails of phospholipids. Well, not only that they are separate parts, they behave very differently when it comes to the water. The head is known as being hydrophilic, so it's attracted to the water. It loves to be close to the water. Whereas the tails are described as being hydrophobic. So it does everything on its power to get away from water, so it wouldn't be mixed with water. Similarly, when you have a phobia as a human being, you try to get away from the source of that phobia. So you might wonder that, oh, that's all very good, but why do I care? Well, it turns out that this difference between head and tails and the relationship between head and tails in relation to the water is key in how phospholipid bilayers are formed, how cell membranes are formed. And this all happens spontaneously in just on its own. We don't need to do anything. You will just end up noticing if you have a bunch of phospholipids floating around, If you put them in water, they will form cell membranes. So now it comes to that why is that then important to us? You remember when we talked really early on this course about how water was so essential to all life, and we saw that most of us human beings were made of water as well. So there's going to be plenty of chances for these phospholipids to arrange in the human body to form these cell membranes because of the rich amount of water that we have present. So that's really the big take-home message that I want to highlight there. Let's have a look of how these phospholipids will now spontaneously arrange in water. Well, I mentioned that these phospholipids will try to get away, so that they are trying to arrange in a way that the tails are always away from the water. If there's water everywhere that we see on this slide, the only way that the tails can get away from water is by packing against other tails. So that's why we end up with this sandwich-like structure, that the tails are facing towards inside of the cell membrane, and the heads, which like water, they're facing outside. So cell membranes are usually kind of these flat sheets. Of course, it doesn't always have to be just a flat sheet. They can be much smaller structures. They can form these kind of little balls or little sacs that still have water inside it, but it's not quite as large as we would have in a cell, a full-size cell. Or they can form these little balls where there's nothing inside, nothing else than the tails of the phospholipids. The lipid tails are facing inside. So all this happens spontaneously without any involvement from us, just because of the way that the heads and tails of the phospholipids behave when mixed with water. So we end up finding many different shaped structures that result as a result. The key thing here that why I included this slide again was to highlight that this all happens naturally, spontaneously. We don't interfere with anything. This is just what happens naturally. The other thing to notice is that not only this gives rise to the cell membranes that we saw when we looked at the layers that surround the cell itself, so forming this outside layer of the cell. This arrangement that we end up seeing also is responsible for all these compartments within the cell. So all these cell organelles that we talked about. So if the phospholipids wouldn't behave in the way that they do, we wouldn't end up finding all these cell structures within the cell either. So really fundamental concept that has enabled for a lot more complex biology to take place. And we'll talk about that more complex biology later in coming weeks on this course, how different cell organelles are so key structures. Now I want to move away from the phospholipids, which were the key component of the cell membranes. And I want to introduce something else. And we haven't, I feel, really spent enough time on highlighting how important these proteins that are embedded into the cell walls, how important they are. And there's two types of proteins that I want to discuss about. The first ones of these are going to be integral proteins. So integral proteins are these proteins that are embedded fully to the cell wall. So they go through the entire width of the, or thickness of the cell membrane. The other kinds, peripheral proteins, they are found only at the extremities. They don't go through completely. So they're all either on the inside or outside of the cell wall, but don't go fully through them. So that's the big thing to remember, those terms: peripheral, referring to these, where they are only partially within the cell wall, and integral proteins going all the way through. Now what I want to talk about is that we should look at the proteins a little bit more in detail on their own. And like everything in life, proteins can be studied in many different levels of attention. So let's start by looking at 3 different key levels of how proteins can be studied. The primary structure looks at the proteins as simply a chain made of amino acids. And you remember that it was the amino acids that we made when we translated the RNA into something useful. So each of these little elements that you see here is one amino acid that has been placed after another. So when we're studying proteins, we can study it on its most simple level as this chain made of amino acids, how they are packed one after another, what the arrangement is, what sort of effects it will have on its both physiological properties, but also on its structure. So this primary structure that we can use to study proteins is nothing but this chain of amino acids. When we go to a little higher level, so take a step away and look at things in a bigger scale, we get to the secondary structure of proteins. And what we're starting to see that rather than just listing individual amino acids, now we're starting to see that these amino acids are taking more 3-dimensional shapes. So we can see amino acids taking shapes as different kinds of sheets, if you wish, or different kinds of the spiral structures, but these are not in 3-dimensional shape. It's no longer just simply a chain of amino acids, but by looking at these in a little bit, taking a step backwards and in a little bit bigger of a scale, we end up noticing that the proteins also have this 3-dimensionality for their appearance. And the tertiary structure is once again a little bit further away. And what we see when we look at tertiary structure of proteins, these might be the ones that you probably see most often when we're looking at proteins. So we're seeing them as these long tube-like structures that are then folding. And this is an important thing for us to notice. And the reason why it's an important thing is that this folding of a protein happens always in the same way if it has particular chemical properties. So by having certain amino acids arranged in a chain, they will always take this particular shape. Well, we learned early on this course that we can look at different things, different molecules in terms of their chemical structure. And by changing the chemistry just a little bit, the entire shape of that molecule can change. And that's exactly what we can do for proteins as well. So again, the chemical structure defines the physical shape of that protein. And if we now start messing with the chemical structure of a protein, the physical shape of the protein will change as well. And this might be a little bit abstract, that what does he really mean and why do I care? Well, why do you care? There's many levels of detail that I can provide you to that. But let's keep it the most simple. You care because this is a question I love to ask in an exam. And I've been teaching for a while, and when I first started teaching, whenever I was talking about proteins to my students, the textbooks tended to have a picture of Nicole Kidman. And I was like, what does Nicole Kidman have to do with proteins? Why is she always there? Well, maybe this is aging me a little bit, so I'm giving it away. At that time, Nicole Kidman was probably the biggest star in Hollywood. She was the it girl. And at that time, Nicole Kidman was known for having straight hair, but she then went and got a perm, and her hair turned into this all curly and all that. So the reason why her picture was on these textbooks and the reason why Nicole Kidman relates to the proteins in this context is that, of course, hair is made of protein. And what she did by taking perm was that she was altering chemically the structure of her hair, the protein hair, and by changing the chemical properties of the hair, also the physical shape of the hair changed. So that's all I want you to know about Nicole Kidman. For some reason, my students get really frustrated, but I still do sometimes just to test whether you've been in a class or whether you watched any of the lecture videos. I do sometimes ask my students that in an exam that, what does Nicole Kidman have to do with proteins? So now that you've been here today, you know that what Nicole Kidman has to do with proteins, as we've discussed in a class, is that hair is made of protein. And once you are changing the chemical structure of a protein, as she did by taking perm, the physical shape of the protein will also change. So chemistry affects the physical appearance of a molecule as well. So I think that even though that is a silly example of this three-dimensional nature of the proteins, it is a really good question in finding out who's actually been in a class and who hasn't. So I'm just putting it out there. I'm not promising, but sometimes I do like to ask that in a test. And if you are totally confused, I do cover this same topic also in our lecture videos discussing Nicole Kidman and her hair. Well, we've seen this part of my PowerPoint already earlier on. So we saw that some of the proteins were referred as being integral proteins. So these were the ones that were fully within the plasma membrane. Others were known as peripheral proteins, so they were only on the external or internal surface of the plasma membrane. So that's usually where I finish off with my students the first part of this lecture. And I think this is a really good chance for us to take like a check that, do you have any questions about anything at all that I've shared so far today as it relates to the chapter material? Because I know that we do go through quite a lot. Normally, these classes that we're doing, if we were in face-to-face format, I would probably spend twice the amount of time that we end up spending together in face-to-face format with my students. So we have time to discuss in much greater detail about all of the little things. So what you're getting is basically like a really condensed key points to note, but not enough to know every little minute fact. And sometimes when you don't know those little minute facts, it gets harder to understand what we're talking about. So at this point, does anyone have any questions about anything that we've discussed? Is there anything that's confusing you or concerning you? What I do want to say, that rest assured, before the exam, I always make sure that you know what to expect. There are no curveballs. The biggest curveball I've ever thrown probably is asking about Nicole Kidman. And now I've really told you that I will probably ask about her. So if you have questions, just unmute yourself or type into the chat. If I don't see questions coming through, I'm just going to soldier on and cover the second portion of our lecture material. So the second portion takes everything we've learned so far and now focuses rather than on the anatomy, which was kind of really the structure that we discussed the first part. Now we focus on the physiology, so the functioning of things. So let's have a look of not just naming things and what things are made, but let's actually give it a little bit more meaning by seeing how all of these characteristics affect now the membranes that we find in the body. And I should be sharing my screen again with you. If for some reason you're not seeing it, let me know. But if I don't hear anything, I assume that we're good. So on this section of the class, I want to be talking with you a little bit about the movement of the materials across cell membranes. And one of the terms that I mentioned at the very early part of the class, I think, was that the plasma membranes are very selective about what is allowed to pass through it and what is not allowed to pass through it. And really, this is what life requires. So for life to exist, we need to have these membranes that let certain substances in and keep other substances out. And that's really the very fundamental basis of what all life is based on. So we're going to be looking at what sort of things are able to cross membranes and what sort of things are not, and what sort of consequences it will have. But as we do so, remember that this is all, really everything that we discuss on this course is about that this is what life requires. For life to exist, we need to have this characteristic for the cell membranes. And the transport of materials, which is really the big topic that we're focusing on, can be facilitated in two ways. We can have passive transport and active transport. And let's start with the passive transport. When we talk about passive transport of materials across a cell membrane, there is no energy being used. This just happens naturally or spontaneously, if you wish. So there is no energy being put in for this to happen. It just happens on its own. So that's why we describe it as being passive. And the reason why this transport happens passively is that you might remember very early on, we've talked a little bit about that sometimes some materials are in greater amounts in certain areas of the cell or certain areas of life, certain areas of the universe where we live. And in certain parts, there's a little less of that. So there is a concentration difference between materials in universe. So there are areas where there's high amount of something, and then there's areas where there's a low amount of something. And what we end up noticing happening in world, just passively, without us needing to put in any extra energy, is that these two even out over time. So wherever we initially had a high amount of substance, eventually that substance travels to areas of low amount until the concentration, until the amount of these substances is equal. And the way we can think of this is that if you take a flask of water and you put a food dye into a particular area there. On this picture, it's this area where I have high written there. So there's a high amount of food coloring on this area. Just passively, without us putting in any extra effort, just spontaneously how life is. What happens is that this food dye travels until the concentration on all parts of this flask of water is the same for this food coloring. So this is an example of this concentration gradient being the driving force for materials to travel from areas of high concentration to areas of low concentration until eventually the concentration is same on these both areas. So let's have a look at a few different kinds of membrane transport strategies that are passive. But before we do so that we can understand why they are passive, I think we should also talk about what does it mean when we are talking about active transport. In active transport of materials, we are actually investing some energy to move materials from one place to another. And usually the reason why we have to invest energy, not always, but usually, is that we are going against this concentration gradient. So we're trying to move substances from not to the direction where they would normally go, but against of what they would normally passively go. So we really have to invest energy to move them across in a way that wouldn't normally happen if we didn't interfere at all. So in this example, what I've shown is, and you don't see this happening a lot in life when looking at naked eye. If we had some molecules in a flask of water and they were in low amounts in that flask, if we are doing active transport, we would be moving these molecules, not so that they spread evenly everywhere, but so that they condense on a specific area. So we're moving against the concentration gradient. So the big thing here is that this would normally never happen. We have to invest energy so that we get these materials to go against where they would normally go. So as a result, they used energy. And now I said that we're going to go through 2 different ways of how materials move through a plasma membrane. And the first one that we're going to look at is going to be a process known as diffusion. So this is an example of passive transportation of materials. So this happens naturally. We are not investing any extra energy to that. It just happens. And what happens in diffusion is that we have substances that try to just passively reach a concentration balance. So what I've got here is a picture on the right-hand side. And on this picture, we're seeing a flask of water. And this flask of water is separated into 2 by a cell membrane, by a plasma membrane. And only on one side of this plasma membrane, we have a high concentration of particles. On the other side, we have none of the particles. If we have diffusion happening, so now we are letting the materials pass through this plasma membrane, and we're letting that to proceed from areas of high concentration to the areas of low concentration. Well, in this case, that will continue until the concentrations are same on both sides of this membrane. So this particular membrane that we have been looking at is now capable of facilitating diffusion. So it lets materials to pass spontaneously from areas of high concentration to areas of low concentration until the concentration balance is reached. And as I said, notice that this is all passive. We do not have to invest any energy on this. So that's the first kind of transportation that we have discussed. Now, let's look at another type of transportation. And we have the same situation as we had earlier on. This is a passive transportation, but rather than being an example of diffusion, we're now talking about osmosis. And what osmosis is, that we try to even out these concentration differences until they're similar, but now the molecules that are within the substance that we're looking, these molecules actually cannot pass through the plasma membrane. So that generates us a challenge because we want there to be same amount on both sides, but these molecules are not able to go through this membrane. So how can we enable these 2 sides to have a same concentration of this substance that we're studying, the red dots, without actually letting the red dots pass through the membrane? That sounds like a kind of a challenging question, but there's a simple solution. So rather than moving the substances, let's move the water. So in osmosis, again, remember that that is going to be an example of passive transportation, but rather than letting the substances move, we move now water. And we move water until the concentration is same on both sides of this flask that's separated into 2 halves by this cell membrane that we saw. We add more water onto this left side, and we add less water onto this right side. Now we have same concentration of these red dots on both sides. The volume is lesser on the right-hand side, but the concentration is the same. So same volume contains similar amount of these substances. So that's an example of osmosis. And you might be now like, oh my God, how do I remember the difference between our diffusion and osmosis? Remember that in my exams, you are allowed to use your notes. So really taking good notes is worthwhile here. So make sure that you have noted that our diffusion was all about moving these actual molecules across the plasma membrane until we reach the same concentration, so that we move them from areas of high concentration to areas of low concentration. The substances move across. In osmosis, these substances don't get to move across, but water gets to move across, again, until we get an equal concentration on both sides. So materials move so that there's the same concentration on both sides. All of these happen passively. We don't really have to put in any energy for this to happen. One good example of where we see this happening is when we use, for example, fluid therapy in humans. So when you go to a doctor and you're sick enough that you get admitted to a hospital or to a clinic, normally one of the very first things we do stick a drip into your vein. And through this drip, we're able to deliver fluids into your body. The other reason why we do that is that once we have this connection established, it's really easy to put in drugs into that drip, and they get really quickly into your body and into your circulatory system. Well, it cannot be just any old water that we put into your body. It needs to be a specific type of fluid. And this specific type of fluid needs to have just the right amount of right components so that it doesn't mess up what's happening normally in your body. And this brings us to talk about a concept that's known as tonicity. And tonicity is something that's going to be really, really important if you're thinking of heading to a career in healthcare field. So tonicity looks at these concentration differences and what sort of effects it might have. And there's three types of tonicity that we'll discuss. And as always, when you study biology or anything in life, really, try to come up with ways that help you to remember stuff. Even if it's a little silly, if it helps you to remember something, it's a method and strategy that's worth using. So I've tried to come up with ways that I find these easiest to remember. They might not work for you, but I'm throwing out some examples. So again, remember what we're seeing in tonicity is this movement from areas of high concentration to areas of low concentration until the concentration gradient is the same on both sides, until there's the same amount of stuff everywhere. So let's start with hypotonic situation. In hypotonic situation, we have the insides of the cell containing more salts and other solubles than the outside of the cell. And remember, we always try to neutralize this amount, so it would be the same amount on both sides of the cell membrane. But in this case, there's just way more substances inside the cell. Well, what your body will try to do, it will try to neutralize this. And how it's going to try to neutralize this is by pumping more water or letting more water to go into the cell. So basically, we're trying to get the both sides to have the same concentration of salts. And how we do that is that let's add more water inside the cell. Well, cell can accept water, and it's just going to start growing bigger and bigger and bigger, swelling. But it can do so only to a certain level. If we keep pumping more and more and more water, eventually the cell might even end up bursting, because now there's too much water inside it, it can no longer take it, and cell breaks. So that can really literally happen in life. If we were to, for example, to give you water rather than a saline drip, when we put that fluid into your body at the clinic or at the hospital, if it was pure water, that water would start to travel inside the cells and eventually cause those cells to burst. And that would not be good when your cells explode, for obvious reasons. Cells were the simplest form of life that we knew. So how I think of the hypotonic cells is that I think of a hippo. Hippos are these big, large animals. So hypotonic cells, a lot of salt inside, less salt outside, water flows inside the cell swells and eventually bursts. Cell looks a little bit like a hippo. So that would not be a great case. Let's look at when it would be the great case. In isotonic situation, we have the same amount of salts inside and outside the cell. So water does go into the cell, but water also leaves the cell. So the amount is always consistent, so there is no change to the cell's size. It doesn't swell and it doesn't shrink. It remains perfect. And how I think of the isotonic cells, I think of them as cells who say, I so perfect. I don't need to change. I'm perfect as I am. So there's same amount inside and outside of the cell. Some water will come in, but some water will leave. So the overall size of the cell remains consistent. Those were our isotonic cells. And the final situation is going to be hypertonic conditions. In hypertonic condition, we have more salts. If we use the salts as an example again, we have more salts outside the cell than we have inside the cell. As a result, again, passively, your body will try to even out the concentrations inside and outside. And as a result, water gets pumped out from the cell because we are trying to even out this concentration. So water is flowing out, and eventually the cell will just shrink and it might even end up drying up. I think of this like as a grape, and when you leave it to the sun, it turns into a raisin. So the fluids travel out from there. So how would you remember this? Well, again, the word hypertonic, I think of what kind of people are those when we describe that someone is very hyper. People that are hyper are very active. They have a lot of energy. And what do you do if you have a lot of energy? You run around like kids that are hyper. Well, if you run around, what happens? You become skinny because you're burning all that energy. So hypertonic cells, to me, in my mind, are the skinny cells. So they shrink and eventually dry up. So this is a good way of thinking of the tonicity. So remember our hypotonic cells, the hippo cells, they were getting water flowing inside. They became big and large and eventually might even burst. So it's not good. Our hypertonic cells, they were losing water, they were drying up and might eventually just literally dry out of water. That's not good. The only time when things are going good is when isoperfect and the cells are isotonic. So there's the same amount of water coming in and going out. There's the same amount of salts inside and outside. You might ask me that, okay, nice. Now there's another thing that I have to know, and I really don't understand, why do I care? Well, we see this happening, for example, with the plants. So what we use to water plants actually matters, whether we are using salty water or whether we're using fresh water. So even though you water a plant, if you use very salty water, that will only lead into water escaping from the plant cells to outside. So you can pour all the water you have, but the plant will just dry, dry, dry. This is the reason why you should not drink if you're on a life raft and you're really thirsty on an ocean and you really just want to get something to drink, but only water that you have around you is going to be salty seawater. You don't want to be drinking that salty seawater because it will just dry you up even more. So we need fresh water for our plants, but also us as humans. So I guess a good way to think about this, and one of the questions that I love asking from the students is that, can you die out of drinking too much water? So if you just keep drinking tap water and you drink that more and more and more and more, can you die as a result of that? And it turns out you don't have to try out. Someone's done it for you. Back in, I think it was in 2007, when the Wii, the game console, you know, the one that you use to play games, was a new thing. That was like the hottest gadget on the earth. Everyone wanted to get a Wii game console. There was this radio channel, and I'm not going to say in what country because I remember it wrong otherwise. But there was this radio channel where they thought that, oh, we have a fun name for a contents. And in the contents, the viewers or listeners of the radio channel can win this Wii console. Let's call it Hold Your Wii for a Wii. And what they literally did, they challenged their listeners to drink as much water as they could in a very short amount of time without going to use the bathroom. Well, now that you know everything about tonicity, you know that that sounds like a really dumb idea. Well, clearly, these radio DJs had not done basic biology classes, and actually one of the listeners did this and ended up passing away. Because what they did end up making their body hypotonic. So there was water flowing into the cells more and more and more, and eventually the cells of that individual bursted and they ended up passing away. So you can cause trouble for yourself if you just drink water. Your body, though, is in too great of amount in a very short amount of time. Body is great. As long as you are getting too much water, your body will tell you that, oh, I really need to go and pee to get rid of that. So that's one thing that this woman could have done. The other thing that could have been done, we could have used diuretics to actually drain some of those fluids away. We could have done a lot of things, but that was too late. This went too far. So really, that's an important thing to notice in terms of that. The last thing that I want to discuss with you as it relates to the, I would say probably to this transport of materials, let's look at a situation from passive transportation. Last example of passive transportation, where we have substances that move across through this membrane, but they move through a facilitator. So we have a protein of some kind that, rather than these materials having to pass the plasma membrane, they can actually pass through a protein. So these proteins help to move the materials from one side to another until again we end up with same concentration on both sides. And this would still be an example of passive transportation. These proteins don't consume any energy. They're just basically like tunnels for these materials to move across through the plasma membrane. And these, of course, need to be integral proteins. They would need to be proteins that go through all the way from one side of the plasma membrane to the other side. And there's two types of proteins at large that I want us to know. I want us to know that we have something known as channel proteins. These are really just tubes or tunnels, like I said. And then we have carrier proteins. Carrier proteins actually change their own shape to physically carry that material across the membrane. The next type of transport that I want to discuss with you is going to be now, for the first time, an example of active transportation. So now we are using energy to transport materials from one side to another side. And how we describe these is we describe these as pumps. So pumps help us to move materials. We need to have energy for these pumps to work. So what we have here is, for example, a cell, and we have outside and we have inside of the cell. And you'll notice that we have atoms, and some of these atoms are positively charged, and some of them are negatively charged. And in this initial situation, we have the same amount of both positively charged and negatively charged atoms on both sides of the cell membrane. But remember, this is now going to be active transportation. And in active transportation, we are using energy to pump things against the concentration gradient. So we are going against what would make it to be the same. We are generating a difference. And for making the difference, we need to spend energy. How we do this is by using these pumps. So they physically move negatives to one side, positives to other side, and we are generating a difference in the concentrations on both sides of the cell. And you might think of that, oh, that's all very good, but why do I care again? Well, you care because when you get to study about nerve cells, one thing that you'll notice is that nerve cells, which are known as our neurons, they are always negatively charged from inside. So there's more negative charges inside the cell than there are on the outside. And it's actually negative 70 millivolts, that's the charge inside the nerve cell. And having this negative charge is what allows action potential, small electrical currents to pass through our nerve cells and pass messages from one part to another. They really are like electric wires that transport electric signals from one part of the body to another. And those electrical signals are what pass messages. The final example, if I remember correctly, I don't think I have anything else after this, is another type of active transport. And in this type of active transport, we are actually moving a lot of material, or then that material is just too large to pass through the cell membrane on its own. So we need to somehow pass material, but this material, these molecules are too big to pass through the cell membrane as they are. Or alternatively, we have a lot of material and we want to take the time to pass every single thing, every single molecule through the cell membrane. So what we do, we actually end up developing this little indent into the cell membrane where the material enters, and then it pinches off, making this vesicle that carries the material inside the cell. So this is how this material is able to cross the plasma membrane. Then once it's inside the cell, this membrane in this vesicle can just break off, and now the material is inside the cell. So this is the way how we can move a lot of material all at once, or if the material is just too large as a molecule size to enter in. And there's two movements. First of all, material can be going inside the cell. If it's entering into the cell, it's known as endocytosis. So that's going into the cell, as we saw in this picture, going from out to inside of the cell. So entering, I again use my mnemonic entering, endo. And if material is leaving the cell, so it's going out from inside the cell to outside the cell, using the same method, but in reverse order, we're talking now about exocytosis. So exo, exiting the cell, that's our exocytosis. Either moving a lot of material all at once or moving material that's too large to pass through the cell membrane. And these both were examples of active transport, transport where we do need to have energy invested for this transport to happen. And that really sums up everything that I wanted to discuss today. I know it was a lot of material, and I'm going to be around if you have any questions about that. Remember that you also have very good lecture videos that I prepared that take a little bit more time than we have a chance on our classes to discuss these processes. And I'm always happy to make the time. I know that I've been a little unwell, so I haven't been able to answer to every message, but if you have any questions, I will definitely answer. Was there a question? I see that there's a chat message, yes, and question mark. Was there a question that you wanted to ask? Well, okay, thank you for answering the question. I'm sorry that I missed your answer. Like I said, when I share my screen, I lose the chat. I really appreciate that you were so actively involved on this class. This is all that I have planned for today's class. What I would like you to do before you leave, if you don't mind again, just post your name on the chat. That way I can take the attendance once more. I take it at the beginning of the class and I take it at the end. It's okay if you can only do one of the attendances, but if you do both, I usually, again, give extra credit for that. This is all I have to share for tonight's session. If you have questions, I will be around. But if you feel comfortable with the material, this is it. I'm going to wish you a very good rest of the evening. We will have a session on Wednesday evening, and we will, oh, thank you. And we will go through the lab. This week's lab is looking at different membranes. And as a model of a membrane, we will use actually an egg membrane. So you would take an egg and you would remove that shell and you would be left like with a huge shell, or a huge cell, I mean. And some materials can enter into that cell and some can't. And we will have a look at that. You can do that lab yourself, but I'm not expecting you to do it. So I will show you the results, what happens when you do that, and we will discuss that on our Wednesday session. So I hope you have a very good rest of the night. Remember, I'm here to help you. Just give me a couple of more days to catch up with any questions that have been mailed because I have been a little sick. And I hope you have a very good rest of the night. I'll be here if you have questions, but that's it for the night. Thank you. Have a good night.